Thank you. It goes this way. So GenSight Biologics is a Paris-based gene therapy company, and we're actually focusing on two different platforms with two strong development for indications. Just try to make this working. Oops. Which one? Okay. Oops, sorry. So the two proprietary technology we are developing are only focusing in ophthalmology today, but you will see there's other opportunities we intend to actually develop later in the future. And the primary indications, well, I would say platform, is a mitochondrial targeting sequence. We'll still have in our genomes 13 major genes that are still expressed in the mitochondria, and that's very important because they have major consequences, and you will see for one specific indications, we could actually have the opportunity to get, oh, sorry for that, to get some disease of specifically the retina, but other central nervous system application. The second platform of development is the optogenetic programs, and we have actually ready to initiate some gene therapy for ophthalmology applications up to the other extension for degenerative disease of the retina. So let's have a look at the first mitochondrial targeting sequence. Basically, Leber hereditary optic neuropathy is a very specific disease of retinal ganglion cells in the retina, and it's an extremely brutal disease. We can consider clearly it's a neurodegenerative disease in a sense that these disease will actually lead to a very rapid and brutal loss of vision in mostly patients who are 15 to 30 years old. And these patients will actually lose vision. In 50% of the case, they will lose both eyes in less than 2 months. In the same time, you will see they have in parallel some very strong retinal ganglion cells disappearance basically, and as it is neuronal cells, there is no way they were going to be able to regenerate the cells. So blindness will occur clearly in 70 to 80% in 6 months in both eyes of the patients, but in the meantime, after 1 year follow-up, this patient will actually, in 98% of the cases, have less than 2400 in visual acuity. It's clear that this sequential loss of vision is also an opportunity for a very rapid development, and that's why we were going to explore in the meantime. In terms of epidemiology, here we're talking about, in terms of incidence, about 1,500 early trained patients, and there's 3 major mutations, which actually the mutation we're targeting is the most severe and focus of about 75% of the patients in Europe and US and about 90% in Asia. Interestingly, the most important part here is that you have to deliver the expression of the protein inside the mitochondria. And this is where the technology actually is being developed by the INSERM unit in Paris, and it's been applied very prudently and very optimally in terms of a demonstration in a rat model and also expression of the in the monkey during the tox program. We could identify that. Very importantly, you deliver pretty classically your cargo and the AAV in the nucleus of the cells, but more importantly, you have 2 major mitochondrial targeting sequence, 1 and 2, which have extremely important role. The first one is going to be able to actually target the polysome at the membrane of the mitochondria, and the second one is to allow the translocation inside the mitochondria to get the expression of the protein. And this is typical of the allotopic expression. And this is where you have the ability to restore the expression of the protein, and we've clearly demonstrated that in animal models, but also in vitro through the potency assay, the ability to regenerate the ATP production, but also the ability to make cells survive very significantly. Now we've been through the normal, I would say, regulatory path, and we actually reached the phase 1/2A study. And after 48 weeks follow-up in different dose ranging, escalating dose, basically, we treated different type of patients. But more importantly, we know as a neurodegenerative disease that you need to treat as early as possible. Clearly, we know that in stroke, for example, time is brain. Here, time is vision for these patients. So what we did is we anticipated the analysis of this study by separating the patient who are less than 12 months from onset of disease and more than 12 months of ascendant disease. And we can clearly see that after 48 weeks follow-up, in patients more than 12 months of onset of vision loss, we clearly couldn't see any benefit in these patients. In the meantime, we could see that we have up to 70 letter difference between the treated and untreated eye, and we treated actually the worst eye. And this benefit is really translated for the patients in not only visual acuity improvement, also in color and contrast improvement. And we try to illustrate this benefit through subjective patient report and discussion, intense discussion between the investigator and the patients to be able to understand what it means for the patient in terms of activity daily living. This patient could see color traffic, for example, traffic lights. They could actually see much better letter, but if they want to read an iPhone, they're going to be close to as 1 or 2 centimeters. And this is a very significant improvement for them. So knowing this data, we actually had regulatory discussion on both sides of the ocean, and we decided to go into phase 3, which have been initiated in the late quarter of 2015. And now we're conducting this study in Europe, France, UK, Germany, and Italy, but also in the US with 3 major centers, notably in Philadelphia with the Massachusetts Pioneer, and also with Nancy Newman, who is the president of the North American Neuro-Ophthalmology Society at Emory, and in Los Angeles with Duane University. And the design is really exploring this issue of bilateral loss of vision. So we are going to treat one eye, and we do a sham procedure in the other eye, not doing a real placebo injection, but a sham. And the primary comparison will actually compare the evolution of the visual acuity, because we know that 98% of the patient will have less than 2400 in both eyes after 1 year follow-up. We currently have treated more than 21 patients, as indicated in the slides, and we clearly expect to finalize the recruitment of the studies early 17 and to be able to get a readout early 18. One key point about these 2 studies is that we have separated the population based on the early assumptions that there's a potential time importance considering the neurodegenerative status of these patients and of the retinal ganglion cells. So we target patients between 0 and 6 months of vision loss, but also between 6 to 12 months. So we're going to get a very homogeneous population on both studies, and we'll be able to do, of course, a meta-analysis to measure whether or not there is a time interaction for these patients. The second platform we're developing as optogenetic is really targeting much more a significant number of patients. Here we really have the retinitis pigmentosa patient, but also the dry AMD. You've seen and you know that retinitis pigmentosa in ophthalmology is one of the most prevalent orphan disease, and it actually leads by more than 100 different mutations. So it's going to be very difficult to do gene therapy globally for this patient, especially on the very late phase of the disease, and motion patients or light perception or bare light perception patients. Here the number of patients are much more significant. We're talking about more than 15 to 20,000 new patients every year, and globally these patients in terms of prevalence are more than 300,000. The other dry AMD is for the late phase, especially for the geographic atrophy. These patients actually have very severe vision loss, and that could be also a very significant improvement if they could gain and reduce the ability to lose vision. So what is optogenetic? The principle of optogenetic is, I could say in a very simple words, to transform the loss of cone and rods, and as is normal structure in the retina of these patients, but actually to use the retinal ganglion cells who are going to conduct the neurotransmission of the electrochemical signal that we all receive into the through the retina, and transform the retinal ganglion cells to become photoreceptor. So for this, we had established some collaboration with the MIT Friedrich Miescher Institute in Switzerland with Bertrand Roska and the MIT group of Ed Boden. So the principle is really to restore and insert the photosensitive protein, which mimic very closely what's happening in the normal situation, and insert this into retinal ganglion cells, but you have to provide the energy and the photosensitivity, photostimulation, and the device to actually restore the ability to analyze the patient vision. So the retinal output you can get really is the photostimulation of the retinal ganglion cells to the brain, to the visual cortex for the interpretation of the patients. So if we try to illustrate this, you can clearly see that after intravitreal injections, because we are targeting the perifoveal retinal ganglion cells, you don't need to go subretinal, which you would need if you were targeting the cone or the rod photoreceptors, basically, is really to be able, by photostimulating and I would say cell by cell or pixel by pixel, you're going to get with a specific camera, you're going to get some micromirror which will photostimulate your retinal ganglion cells, and we're going to get a programming which will explore and encode this image into the brain of the patients. The stimulation of the optoelectronic device is an external device specifically designed as this very sensitive red light, because if you use blue light, there's some discussion about the potential phototoxicity associated with blue light. So how do you demonstrate that this kind of optogenetic program can actually photostimulate and be really a reality? What we did is, of course, we did a lot of mice and rat explorations, and also demonstrated clearly some, I would say, neurocomputer changes from the rat. But the most important was to look at the monkey, which mimic much more the retina of the human patients. So what we did is we did a dose relationship analysis, and when you look at these pictures, you can clearly see that you can fire actually the different ganglion cells of the monkey, not only with a dose response of the light stimulation, but also a dose response of the vector and the protein we have expressed. So that's a very, I would say, highly motivating perspectives, because optogenetic can lead to different opportunities. And this program is going to enter into phase 1 next year. And the readout is pretty straightforward, because you use generally patients who have very low vision, to say, and motion to no light perception. So any benefit you can get from these patients through motility or through mobility and through activity daily living is pretty straightforward. So as a conclusion, if we think about the mitochondrial targeting sequence opportunity, clearly we could see this mitochondrial platform could not only target one mutation of the ND4, which is the Leber hereditary optic neuropathy, but also the dominant optic atrophy, but also other mitochondrial disease that have major consequences, like the NOP or the leak syndrome, but also in ALS, and based on different experimental models we are conducting. At the same vein, optogenetic platform has a very high potential, notably in the sensorial and non-sensorial. But for example, if we think about deep brain stimulation, or if we think about congenital deafness, it's a very significant opportunity once we have a clearly established optogenetic as a high value for clinical translation. So this platform, and we intend to leverage significantly this expertise, not only in the pipeline of ophthalmology, but also in the pipeline of neurodegenerative diseases. Thank you for your attention.